Now the stroma of a chloroplast is not an air-filled space. It's a fluid-filled space, a very thick fluid filled with enzymes. These are all metabolic pathways we're talking about. Each one of these run or driven by enzymes. But we need these products out here from the light-dependent reaction to use in the light-independent reaction. Now the reaction that's done in the stroma is a cycle, and there are several steps in this cycle. The first being you've got to incorporate carbon dioxide, because the carbon dioxide eventually becomes the glucose. And where does carbon dioxide come from? Yeah, we exhale it. Yeah, we're making it when we're burning sugar, and we'll learn about that during cellular respiration. But it's in our atmosphere. So plants get it from the atmosphere, and they either immediately will use it in the stroma, or there are some plants that will store it. There's other pathways that will store it and then eventually use it in this pathway. This is the Kelvin-Benson pathway, or the C3 pathway. Because one of the end products that's coming out of this is a 3-carbon compound that when you put 2 of these together, you're going to get glucose. So, and that's this molecule right here. So we got to see this being made, but we're going to show how we're going to use the products from this light-dependent reaction. So carbon dioxide comes in from the atmosphere, and it's either stored or immediately used, depending on which plant it is. And there's a compound here called ribulose biphosphate, or we're just going to call it RUBP. And that is a 5-carbon compound. So when you get this 1 carbon in CO2 with the 5 carbons, what's 5 plus 1? You want me to get you a calculator?
6.
Okay. You had to use your second hand for that one, didn't you? So 5 plus 1 is 6. This ends up being a 6-carbon compound. There is an enzyme that catalyzes that reaction, considered one of the most abundant proteins in the world, because enzymes are proteins. And its abbreviation is Rubisco. And we know that enzymes have their suffix ends in ASE, so why doesn't Rubisco end in ASE? Well, it's because that's the abbreviation for the actual really long name. I couldn't even write on the board. No, I probably could, but this is ribulose biphosphate and carbon dioxide. The enzyme that catalyzes the reaction to attach those 2 together to fixate the carbon dioxide to the RUBP is ribulose biphosphate carboxylase. And you just need to know it as Rubisco. So when you attach these 2, you get a 6-carbon compound. But this 6-carbon compound is so unstable that they can't even isolate it. It immediately breaks up into 2 3-carbon compounds. And these are PGA, phosphoglycerate. Oops. And notice how we haven't even used any of these yet. We've just needed to, and where's this happening?
Stroma.
Out in the stroma of the chloroplast. And where did this happen? On the thylakoid membrane. So this has to happen so we can have these products to drive this second half reaction of photosynthesis. So now is when, let me get some of this messiness off here. Now is when we're going to use some of these products from the light reaction. An ATP molecule will phosphorylate the PGA, so it adds a phosphate group to it. And then it will also, the NADPH becomes oxidized, reducing the PGA. So the adding of the phosphate group and the adding of hydrogen ions and electrons to the PGA now makes it phosphoglyceraldehyde, or PGAL. And there is a more organic name than phosphoglyceraldehyde, which the textbook will talk about. But you'll see the PGAL is the abbreviation. This is a 3-carbon compound. And when you get 2 of these 3 carbon compounds together, that's when you make your glucose. So the carbon dioxide comes in, and there it is coming in, but there's a lot going on before you actually get this glucose made. And because this is a cycle, the textbook will talk about how this cycle has to turn so many times in order to regenerate this RUBP. Because if you just make this and then this is gone, then you won't be able to make any product at all. So this has to continually recycle because you've got a 5 carbon compound eventually making 3 carbon compound. So you have to have this regeneration, and then like the 6th turn of this cycle there's going to be extra PGAL. So it's got to turn again to regenerate this, and then the extra, you'll have extra PGAL, and you put 2 PGALs together, you get a glucose. Well, this regeneration to switch the PGAL to the RUBP when you're turning this cycle is going to use another ATP. You got to phosphorylate the ATP. So once ATP loses a phosphate group, what's it become? ADP. So anytime you see this ATP coming in, all it's doing is losing a phosphate group and some energy is being used to drive the reaction. So when you look at this, let's erase some of this stuff around here. Whole photosynthetic equation, and again, this is not balanced. You've got the water molecule and carbon dioxide. I mean, they're used in 2 different areas of the chloroplast. The water molecule is used over here in the thylakoid membrane to regenerate some electrons. And the carbon dioxide is used over here in the stroma. So it's not like you're taking carbon dioxide and water and putting them together and you got sugar. You've got a whole series of many different metabolic pathways occurring. The end result is glucose being produced and oxygen as waste. And there was a time when this reaction didn't occur. There were other hydrogen sulfide reactions, chemosynthesis producing some energy, very primitive times. And then when photosynthesis evolved, oxygen started being liberated, and we actually know in the Earth's history of our the stratification of our rocks, when rocks begin to be oxidized because oxygen became liberated from photosynthesis and started accumulating in the atmosphere. So thank goodness this occurred, because that's what we need when we breathe in. Otherwise, it was a non-oxygenated atmosphere. There are 2 other pathways. The C4 pathway, which some plants use like corn, and there's another enzyme that's going to fix carbon dioxide to hold it in the leaf, but then it will hand it off into the C3 pathway once these are being produced. Why would you need an alternate pathway? This C3 pathway is in cool, moist environments.